For over 3 decades, the study of Alzheimer's disease has been governed by a single monolithic premise, the amyloid cascade hypothesis. This model dictates that the accumulation of insoluble amyloid-beta plaques is the primary engine of neurodegeneration. But clinical outcomes paint a contradictory picture. We consistently observe resilient individuals harboring massive amyloid plaque pathology who exhibit absolutely no signs of clinical dementia. This scatterplot maps plaque burden on the x-axis against cognitive decline on the y-axis. Notice the outliers, patients with severe amyloidosis maintaining normal cognition. Changing the variable to soluble amyloid-beta 42 reveals a predictive correlation. High A-beta 42 preserves cognition. This clinical data table shows the limitations of the latest FDA-approved monoclonal antibodies, like lecanemab. The metrics reveal that while these drugs are highly effective at clearing fibrillar amyloid from the brain, they only yield a modest 27% slowing in cognitive decline. Clearing the physical plaques does not arrest the progression of the disease. The existence of plaque-resilient individuals and the incomplete efficacy of monotherapies confirm that Alzheimer's etiology is more complex than a linear consequence of fibrillar toxicity. This persistent discrepancy necessitates a rigorous reevaluation of the disease mechanics. Researchers Zhen Huang's competitive synaptic plasticity hypothesis provides a new framework, reframing the function of amyloid-beta in the central nervous system. This model identifies Alzheimer's as a protonopenia, a disease driven by the lethal functional depletion of soluble amyloid-beta monomers. This requires discarding the assumption that amyloid-beta is a biological error or a useless metabolic byproduct. It is an ancient, highly conserved, and active neuromodulator that the mammalian brain relies on for daily neural circuit function. To understand the mechanical collapse observed in Alzheimer's disease, we have to map the healthy, physiological biochemical grammar of competitive synaptic plasticity. We can trace the evolutionary origins of amyloid-beta to ancient bacterial antimicrobial peptides, specifically observing the quorum-sensing behavior of proteins like nisin. Nisin operates with a biphasic, concentration-dependent duality. At low population densities, bacteria secrete nisin as a localized protective monomer. But as population density rises and resources drop, the peptide aggregates into highly toxic, membrane-spanning oligomers to destroy competing cells. The mammalian central nervous system utilizes an identical two-layered cloud mechanism. A highly active presynaptic terminal secretes amyloid-beta in a dynamic concentration gradient, producing a tight cloud of protective monomers that diffuse outward and cross-link into dense oligomeric clusters at the periphery. At picomolar concentrations, that immediate monomeric cloud provides intense neurotrophic support. It physically enhances the recycling pool of synaptic vesicles and lowers the threshold required for long-term potentiation. By the time those peptides diffuse to less active neighboring synapses, they have concentrated into the high nanomolar range. These cross-linked oligomers suppress calcium-triggered exocytosis and facilitate long-term depression, actively weakening the competing connection. This biphasic, concentration-dependent amyloid-beta gradient is the foundational mechanism the healthy brain uses to constantly evaluate and scale the relative utility of its synaptic connections. For this gradient to dictate structural changes, individual synapses must possess intrinsic fitness sensors to interpret the signals and determine which connections survive. The brain achieves this via the flower protein fitness checkpoint. Active synapses, bathed in their own protective monomeric clouds, express the dominant flower Win isoform on their surface receptors. Adjacent suboptimal synapses, subjected to the oligomeric stress signal, undergo a distinct conformational shift. Oligomers bind to high-affinity receptors like PRB, forcing the weaker synapse to express the flower-lose isoform. This flower-lose tag initiates the execution phase. It induces the externalization of phosphatidylserine on the synaptic membrane, creating a universally recognized eat-me signal for the brain's innate immune system. Homeostatic synaptic pruning relies entirely on this continuous, localized translation of amyloid-beta diffusion gradients into discrete flower isoform expressions. This competitive plasticity system carries an inherent danger. If the brain's resident immune cells, the microglia, are not strictly and continuously restrained, targeted physiological pruning rapidly escalates into rampant synaptic stripping. Data published in 2024 reveals how this restraint operates. Soluble amyloid-beta monomers serve as the constitutive physiological brake on microglia by binding directly to the microglial amyloid precursor protein, or APP, receptor. This binding activates RIC8A, a guanine nucleotide exchange factor, alongside a heterotrimeric G-alpha subunit. Together, they trigger a potent anti-inflammatory cascade that halts the production of inflammatory cytokines. Rigorous genetic testing validates this circuit. When researchers engineered conditional murine knockouts, specifically deleting the genes for RIC8A or AP within microglia lineages, they directly dismantled this protective pathway. The resulting cellular phenotype is catastrophic. Without the monomeric brake, microglia lose all homeostatic control. They become constitutively hyperactive, hypersecreting massive amounts of inflammatory markers like interleukin-6 and TNF-alpha, regardless of any external immune stimuli. Soluble amyloid-beta monomers provide the absolute constitutive inhibitory signal required to prevent the innate immune system from consuming the neural network. The pathological state of Alzheimer's begins when genetic or metabolic shifts alter the amyloid-beta 42:40 ratio. This heavily favors rapid hydrophobic self-association. This runaway aggregation acts as a massive thermodynamic sink. As fibrillar plaques grow exponentially, they relentlessly draw from the interstitial fluid, causing the concentration of free soluble monomers to plummet well below the baseline physiological threshold. The immediate consequence of this proteinopenic state is mechanical. The microglial APP RIC8A brake is physically starved of its monomeric ligand, completely disinhibiting the innate immune system. Disinhibited microglia flood the local environment with natrix metalloproteinase 9. This enzyme aggressively cleaves and dissolves the protective perisynaptic extrasynaptic matrix, leaving mature synapses structurally exposed. A violent cytokine storm follows. Massive elevations of IL-1 beta and TNF-alpha bombard the unprotected neurons, directly stimulating intracellular p38 MAP kinase and CDK5 signaling pathways. These hyperactive kinases aberrantly phosphorylate the tau protein. As tau detaches, the structural microtubules crumble, causing the proteins to aggregate into neurofibrillary tangles and triggering total axonal collapse. In this framework, tauopathy and neuroinflammation emerge as the direct, mechanical, and inevitable consequences of amyloid-beta monomer depletion. The monomer depletion model necessitates a strict reappraisal of modern FDA-approved anti-amyloid monoclonal therapies. Antibodies like lecanemab and donanemab effectively clear insoluble aggregates. This action successfully reduces the local concentration of the toxic oligomeric punishment signal, but it fails to address the core deficit driving the disease. These therapies do not replenish the depleted monomer pool. Depending on the specific epitope targeting, certain antibodies may inadvertently bind and sequester the brain's remaining scarce soluble monomers, actively worsening the underlying protein-epinic state. Reducing the oligomeric burden provides temporary relief to synaptic stress, yet the underlying microglial hyperactivity cannot be resolved until the vital monomeric neurotrophic signal is restored. The Huang framework redirects therapeutic development away from broad amyloid clearance, aiming instead at highly specific targets designed to restore system homeostasis. The first intervention is monomer restoration. Pharmacological agents, such as small molecule kinetic stabilizers, could be engineered to halt the rapid cascade of amyloid oligomerization, specifically preserving the availability of the soluble monomer pool. A second target is the flower fitness pathway. Developing precise antagonists to mask the flower-lose receptor on synapses would blind microglia to the pathological eat-me signals, halting synaptic stripping during the earliest phases of the disease. A third approach bypasses amyloid dynamics entirely through direct microglial modulation. This involves deploying artificial agonists to re-engage the microglial APP RIC8A receptor, or deploying systemic MMP9 inhibitors to physically protect the neural matrix, regardless of a patient's plaque status. To halt the progressive erasure of cognitive function, the scientific community must focus on actively restoring the foundational thermodynamic balance of competitive synaptic plasticity.